This, like many plants, has been produced by micropropagation, or tissue culture. It's a remarkable fact that in theory, almost all of the millions of cells in this plant could develop into identical clones, or copies of one another. All the genetic information is in the cell and can be regulated or controlled chemically. In practice, only a specialized group of cells, the apical meristem, which lives in here, those are the only ones that are used. And it looks like this. Surrounding the dome are leaf primordia, and at the base of these are axillary meristems. And it's by stimulating the growth of these that many small plantlets can be produced. First, the surface must be sterilized. Then the scale leaves that surround it are carefully teased away. It's only the very tip of the shoot that's wanted, and once removed, it's placed on a jelly-like medium that contains nutrients and growth-promoting hormones. Different plant species have quite different nutrient requirements, so each is tailor-made. Under carefully controlled conditions, this so-called meristem tip will grow into a profusion of small but perfect shoots in just a few weeks. At this stage, each individual shoot can be transferred to produce more shoots, all identical. But so far, none of them have grown roots. By transferring them onto another medium, this time containing root-promoting hormones, the plants will develop and produce their own means of support. As a means of multiplying plant stock, it's very fast indeed. One commercial company is already producing 70,000 plants a week, almost a conveyor belt production. But that's not all. The added bonus is that the plants are virus-free. At the National Vegetable Research Station in Wellesbourne, scientists are using micropropagation techniques to produce virus-free stocks of various crop plants which can then be released to the grower. Dr. David Walkie explains.
Most virus-infected plants show severe symptoms. In the case of marrow infected with cucumber mosaic virus, the reduction in yield and vigor caused by the virus is obvious. And similarly, in bean infected with bean common mosaic virus, you can see the greatly reduced leaf surface, the dark and light green islands caused by the mosaic reaction. Virus-infected plants in the field, unlike bacterially infected or fungal infected plants, cannot be cured by chemical sprays. And therefore, we've had to look for another method to eradicate virus from the infected plant. And we have been fortunate that tissue culture allows us this technique. Now, in the tissue culture technique, we are able to take the meristem tip from the actual bud of an infected plant and grow it on a culture medium to produce a virus-free plant. The secret of virus eradication by meristem tip culture in many virus infections is the virus itself doesn't actually enter the tip of the bud. In some cases, however, virus does enter the meristem, and in these cases one has to combine heat treatment of the parent plant prior to tissue culture in order to eradicate the virus. The heat treatment stops the virus multiplying so that when you cut out the tip, the piece of tissue is virus-free. And it's probable that over the last decade or so, this has probably been the most economically important use of tissue culture that has so far been used in horticulture. We now have to carry out extensive tests to ensure that this plant is actually free of virus. by taking sap samples from this plant and rubbing them on to a highly susceptible test plant, such as Chenopodium quinoa, which is closely related to fat hen, the weed which we know commonly in our own fields. And if there is virus in this plant, then within about a week or so, symptoms will develop on this test plant. Now, if there's no virus and no symptoms develop, then we can safely say after maybe 3 or 4 such tests that this is healthy stock, and we're able to maintain our nucleus of virus-free plants and the virus-free conditions so that aphids and other vectors of the virus are not able to reinfect the virus, and thus we're able to multiply up the plants and return them to the grower.
Such virus-free plants will save the British grower many millions of pounds this year alone. But it's perhaps in plants like these that we'll see a payoff in future years. They are virus-free potatoes, and they're paving the way to a quiet revolution in potato seed production. At the John Innes Research Station, Graham Hussey.
Potatoes are a major crop industry in this country, representing 6 to 7 million tonnes per annum. But they're highly susceptible to virus, which can substantially reduce yield. Virus is transmitted by aphids, by organisms in the soil, or even by contact. And once plants are infected, the virus is passed on to future generations through the seed tubers. So, to obtain maximum yield and quality, growers must start with high-grade seed derived from virus-free stock. With present conventional methods of producing seed, cuttings like these from virus-free plants are planted in pots before transfer to the field. Plants for seed are grown in isolated locations where aphids are scarce on virgin land. Nevertheless, there's still some chance that plants will become infected. Now, it takes 4 years for each single cutting like this to be multiplied to the first 7,000 plants. And during this time, frequent retesting for disease is necessary and any obviously infected plants destroyed. Now, if the original virus-free cutting is multiplied by tissue culture, these 7,000 plants can be produced in only 5 months. And all these are guaranteed virus-free. Tissue culture starts with a sprouted shoot, but instead of rooting this, it is cut up into nodes, each with an axillary bud. After sterilization, these nodes are placed on a nutrient medium, and in a controlled environment, the axillary buds grow up into leafy shoots. After about 4 weeks, individual shoots are similarly cut up into nodes, which produce more shoots. This process is repeated at monthly intervals, the total numbers increasing by a factor of about 8 each time. Unlike many other shoot cultures, potato shoots spontaneously form roots. When required for planting out, the rooted shoots can be washed free of agar and put into small pots for transfer to the field. Now, an alternative method we're developing makes use of miniature tubers. If cultured shoots are left in the vessel under the right conditions for a further 2 months, small tubers about 5 millimeters in diameter are formed. Since shoot production is so rapid, there is plenty of time within 1 season to produce these miniature tubers. Now, these should be much more convenient for planting and much more convenient for transport than conventional cuttings. They could even be pushed into small peat blocks like this, which could be planted directly into the field, and this may even lend itself to mechanical planting. So we're hoping that the development of this method with miniature tubers can significantly improve the present methods of producing virus-free seed.
Micropropagation is equally useful for the pot plant industry. Don Holgate of Twyford Laboratories.
Now all these plants, the Cymbidium, the begonia, the anthurium, and the pot plant anthurium, pentas, and the slipper orchid, have all been developed by micropropagation techniques. The total British pot plant market is anticipated to be about £30 million in value in the current year. But the Dutch value is anticipated for cut flowers and pot plants to be at least 150 million. The average British family spends approximately £3 per year on pot plants and cut flowers, whereas the Dutch and German families will spend in the order of 26 to £30 per year. So there is clearly an opportunity for expansion within the UK market. The high-quality plants which have been on sale within Holland and Germany are now becoming available within the UK, and we have the capability to micropropagate these plants and to produce them within the UK system.
Well, with me here today are two of the people who helped make that film, Dr. Graham Hussey of the John Innes Institute and Dr. Don Holgate of Twyford's. Dr. Holgate, you're at the sharp commercial end of this business. How strong is the demand for tissue culture commercially?
Well, we've been involved for some 16 years in research, development and commercial production, but the majority of our business, and that's well over 95%, has continually been overseas or export business.
Why is that?
Very difficult to say, but primarily the rate of development of the markets, but also in other plantation crops as well, because we've moved from ornamentals into a whole range of plantation crops, both for arid lands and for tropical agriculture as well.
Are there different attitudes then in countries other than in Britain to your sort of work? I mean, do you meet opposition from more vested interests in this country than, say, in Holland?
I think that we are encouraged to work with particularly the Dutch and the German growers, because they are keen to take advantage of all new developments and to get to their market point as fast as possible.
What would you say are the best applications, the most profitable from your point of view?
Well, from our point of view, we have to be successful, and that can only be if the grower is successful. So we must provide him with carefully selected material which has been made free of disease, bacteria, viruses. The plants must be delivered on time, at the right price, of very uniform size, so that the plants can be looked after en masse, almost on a factory system.
And you can get the skilled labour easily enough to do that?
Well, we're fortunate to have excellent staff who are willing to be trained and have proved to be very successful in all aspects of the work.
Dr. Hussey, those tiny potatoes that you showed us, do they have exactly the same, what you might call genetic stability as any other seed potato?
Well, we hope so, and I think this is the crucial point, because as anyone who has propagated plants vegetatively on a large scale knows, all these plants are subject to the occasional mutation, usually off types. And at the John Innes Institute, we've been looking very carefully into the process to make sure that the propagules, that is, are genetically stable.
Can you see the next development commercially from your research work? I mean, is it a straightforward?
Oh yes,
move?
if we can prove that the propagules are genetically stable, there seems to be no limit to the method. Briefly, it's a question of increasing the quality of the product, so you get better yield and quality tubers, and introducing new varieties some 2 to 3 years faster.
Yes, well, let's just break there for a moment, because Dr. Hussey mentioned quality, and in fact, like a lot of these new techniques, it's not all plain sailing. In fact, in some areas, notably in the United States, tissue culture has received a bad reputation, mainly because of problems experienced using so-called callus tissue. We'll talk about this aspect later. Callus tissue is a mass of unspecialized cells produced by growing bits of plants on growth hormones. If callus is subjected to a new chemical stimulus, it'll grow new plants in profusion. In this case, It happens to be celery. Well, could you tell us, what is callus?
Well, callus is formed at the cut or wounded surfaces of plant tissue. When these plants are cut or wounded, hormones are released in the tissue and cells grow out. Now, if these hormones can be incorporated into a rich nutrient medium, callus will grow very vigorously, and so the plant can be propagated as an unorganized mass of cells. Now, some of these cells are capable of producing plantlets.
Is it as serious a limitation as the people in the States appear to say?
Yes, I think so, because callus, unfortunately, although it looks very attractive for producing masses of plants, produces a lot of abnormal cells, genetically abnormal cells. So these could give rise to genetically aberrant plants.
Is it right across the board that this is a problem? I mean, is it a problem for you with your potatoes or not?
No, the main problem with the potatoes, perhaps we ought to point out that the main problem is the type of shoot meristem that's involved. Most of the shoot meristems that we've seen on the film, and particularly the potato, are axillary meristems. Now, these are existing shoot meristems on the plant, and these almost always give rise to plants which are true to type. Now, shoot meristems can also be formed on other parts of the plant, on pieces of leaf or stem, root, or from callus cells that can grow out of these organs. Now, these so-called adventitious shoots are much more susceptible to genetic change.
Couldn't this be an advantage, Dr. Holgate? I mean, couldn't you use callus almost as a mutation or an induced mutation?
Yes, you must understand the axillary bud technique, or this budding technique.
Well, I'm trying to.
Yes, is essential to get uniformity, and this is what we use in our basic propagation procedures. But we can use callus, and indeed it is used for selection of different genetic lines, and it is important for the breeder so that he can select variants which he can then use for future breeding, getting new genotypes being created. It may be that the cells from the callus are polyploid, or in fact that there's some other aberration in it which is of advantage.
Where does the balance of advantage lie then in the two techniques of using tissue culture for multiplication and, in the true sense, and using callus for multiplication of new varieties?
For multiplication purposes, you have almost an immediate saleable product. From the point of view of selecting new varieties, you've got to go through a whole breeding program, selecting out your new variety, using possibly the mutant lines, but then the new variety has to be multiplied up again for rapid application.
How far can the production of callus be controlled then? I tried to follow your explanation.
Well,
but how
in
far
some
can you control
cases, the callus does seem to be genetically stable, but the general rule is that it's unpredictable. It may be very bad, may be genetically very unstable.
Well, until scientists can control callus development, it remains a mystery, which for many reasons is disappointing, because it's perhaps here that we may see great developments in the future. If callus cells are grown in liquid culture, they mimic the phases of embryo development in the seed. This embryoid is truly comparable to the real seed. Scientists at the National Vegetable Research Station have had a degree of success in surrounding these embryoids in a nutrient capsule and fluid drilling direct into the soil. The age of the synthetic seed, perhaps. Well, Dr. Curran, do you agree with their assessment?
Yes, I think it could take some time before callus culture can be used in commerce, but it would depend on how much more work is put into it and how much money is put into it.
Well, as I explained, that program was made predominantly for farmers, but did we miss any techniques that biologists might use?
Oh yes, I think so. Most of the techniques which were shown in the program are fairly well-established techniques, and they're characterized by having a very substantial labor-intensive input. What we tried to do when I was at NVRS was to develop a cheap technique of producing plants en masse with a very low labor input automated as far as possible.
Well, had you reached the stage where this technique was fully developed?
We got a long way down the line. A key part of this process is to get the embryoids to be self-supporting and not in sterile conditions, so that when you put them into the field or the glass house, they won't die. And we were able to do this.
How stable are they? How long can you keep them?
You can keep them for several weeks in the state which we saw them on the film. As
Without
tiny
involving
planters.
too much expense?
Oh yes, simply by letting them stay in culture and be oxygenated. But if you wanted to store for longer periods, there might be problems. We don't think it would be necessary to store them because you can trigger the reaction and get them to grow almost at the drop of a hat.
What about the question of variation? Have you reached the stage where you can reduce it in practice?
No, we haven't. What we felt was that it's essential first to get a cheap technique for producing many plants by this method. When we can do that, then it will be possible to study the variation, as has not been possible in the past. We can use statistical techniques, we'll have plenty of plant material to make the judgments on.
Is the variation very much wider than it is under natural conditions?
I doubt it. With the crops that we were interested in, we were interested in cross-pollinated crops, where there is a great deal of genetic variation anyway. Crops like celery, carrots, parsnip. And in comparison with that, probably there wouldn't be too much. In crops like that, the sources of variation are both genetic and from the mother plant. And we would hope to eliminate both of these sources of variation simply by cloning the material.
And what's the biggest limitation before this becomes a commercial reality?
I think sufficient interest on the research side and the money that goes with it.
What about interest from the commercial, from the growers, from the farmers?
Well, I think once they realised that they could have cloned carrots, cloned parsnips, and things of this nature, then they would be clamouring for it, because at the moment they have to put up with a cross-pollinated crop with a great deal of natural variation.
And you think they'll pay a premium for this particular material?
Almost certainly. They will already pay a premium for specially selected seeds.
Oh, and that's where things stand at the moment. Other countries seem to be pushing on with this research rather faster than we are. And if we don't get the necessary background information, we could find ourselves as seed producers in this country way behind our competitors.